insect tissue culture pioneers are the ones who started this conference. And then, of course, they had to add on the fish cell culturists because of all the advancement going on between the different systems. So anyways, we definitely want to welcome you to the 16th International Conference on Invertebrate and Fish Cell Culture. And we, as being the conveners, I'm Cindy Goodman. We have Vivian Day and Shirley Pompani over there. Thank you. So we all represent 3 different areas of research. Invertebrate would be myself and Shirley, Shirley being the marine invertebrates. I'm an insect cell culturist. And Vivian is our official fish cell culturist. So, of course, we want to thank the sponsors of this international conference. So we have 3 outstanding sponsors, Schmidt Ocean Institute and Cryocrate, LLC, and finally, SIVB itself, the In Vitro Animal Systems section of SIVB has also helped sponsor our speakers, as well as the Schmidt Ocean Institute sponsored our refreshments. And before I forget, the refreshments during break will be in the room next door. So when the break comes around, please feel free to go into the next door room. Something that we're also excited about is that we have a special issue coming up, and we want to invite everybody to submit a paper. This is not just a proceedings, although these international conferences generally have had proceedings. This is a special issue that's open to everyone, not just in the audience, but any of your colleagues that have done any kind of work with invertebrate or fish cell culture. So we're welcoming everyone to consider submitting. The deadline is September 30th of this year. It was recently extended to encourage as many people that are interested in submitting a paper. So the schedule for today, of course, we have the morning session, and we're the functional concepts underlying successful cell tissue culture strategies. And then in the afternoon, after you have a very brief lunch, sorry, it's only an hour, but then we'll get into our second section on current and future applications of invertebrate and fish cell culture systems. Now, we don't want you to run away after that, because there's going to be a very unique roundtable discussion immediately after that second session. And the Society for In Vitro Biology is very unique in the idea that we have a lot of plant as well as animal cell and tissue culturists who come to these meetings. And so we're going to take advantage of that and have this roundtable discussion on media with experts in all these different fields. And so it's going to be very interactive, so come with your questions. I know there's already a question on the table about how we can use AI in optimizing media for cell cultures. So we will have this panel discussion. The panelists will be talking to one another as well as interacting with any kinds of questions that come forward. Just a very, very brief introduction to our session. I wanted to bring out and point out the fact how long tissue culture has been in business. So I was going back to the 1800s. And then as it advanced, Carrel in 1912, that was when they really brought in the whole concept of the importance of sterile technique. And then as we go through, I'm not going to go through all these names are going to be familiar to many, and of course the HeLa cell line came around in 1951, but really not much longer after that, we had the first insect continuous cell cultures being established in 2 different continents, China and Australia at the same time. And then immediately following after that, we had our first fish cell line. So the fish cell culturists and the insect cell culturists were really not very far behind the human cell line cell culturists. And what's also very exciting right now is history is still in the making, because we have Megan Conklin here in the audience who will be speaking this afternoon, and she was the one involved with Shirley Ponpony and her lab who produced the first invertebrate marine cell culture from sponges. And this is very recently, and so this is very cutting edge, and you'll hear from Megan later this afternoon. Okay, with that, I'm going to invite Vivian up to the platform to introduce our first speaker. Thank you very much.
Excellent. Well, thanks very much. We have two individuals that are going to come up and present for our next talk. First off, we'll have Michael Saad, and we'll then have Sophie Letcher, both of which are PhD candidates at Tufts, working sort of within the fish cellular agriculture space. So again, I just want to truncate time and invite both to come on up to wow us a little bit more about fish and insect cultivated meats. So, do you prefer
Hi, everyone. Can you hear? Cool. So I'm Michael. I'm a fourth-year PhD student at Tufts.
And I'm Sophie. I'm also a fourth-year PhD student at Tufts.
And our research focuses a lot on cultivated meat, mine specifically with fish cells and Sophie's with insect cells. And so just to get into a bit of background for the field of cultivated meat, we know that animals provide a lot of tasty, nutritious food, things like sushi, like hot dogs. We also know that animal production has a host of issues with pollution and greenhouse gas emissions, resource utilization, especially land use, health concerns over things like antimicrobial resistance, and animal welfare. And we've known about these issues for a while, but despite that, animal production continues to increase. So you can see over the past few decades that animal production has increased quite drastically. And so because we know of these issues and that people aren't going to stop eating animal products, a number of alternatives have emerged. Most recently, we've seen a boom of plant-based meat with products such as Impossible or Beyond. And while that boom was great 5 years ago, it's now starting to slow down a bit. And when consumers are polled, the reason they cite the most is that the products just don't taste good enough. They don't taste as good as the meat that they're used to. And so while Sophie and I do enjoy those products, we're also interested in presenting more alternatives, and that's where cultivated meat comes in. And so cultivated meat tries to replicate meat by actually focusing on the cells themselves. So meat is composed primarily of muscle and fat tissues, and so this field focuses mainly on muscle and fat tissue engineering. And so here's a diagram just showing an overall process of cultivated meat. You can see that it starts with the cells, either primary cells or immortalized cells, and then there are two different bioprocesses that we generally think of. One is a proliferation bioprocess, and that's where we try to accumulate cell mass, and then the next is a differentiation process. That's where you mature the cells into more meat-relevant phenotypes. And what's important about a constraint of cultivated meat is that because we're trying to reduce animal use in our food system, a lot of the animal-derived products that you're used to using in cell culture won't be used in these processes. So things like FBS are really important to remove from both the proliferation and the differentiation processes. And after we have those mature cell types, the final step after some downstream processing is to take our cells and formulate a product with those cells. And so cultivated meat is a relatively new field, about 10 years old. And when we think about, if we look back at this figure, we see a cow. And most of the research in the field has focused on more mammalian systems, cow and avian systems. And so you can see in this figure right here, which is adapted from the Good Food Institute. It's a nonprofit that focuses on plant and cultivated meat research. You can see that the number of papers published with cultivated meat papers are dominated by cows, pigs, and chickens. And I think that makes sense. These are more analogous to the human and mouse cell lines that biopharma has been growing for a while. And you can see that while they do make up a significant portion of the meat consumption per capita, that's in the tan, the magenta, and the pink bars, that there is still a sizable blue bar in the meat consumption per capita. And and that's fish and seafood. But when we look at our papers database, we see just a handful of fish papers that have been used for cultivated meat. So there are a couple from zebrafish, and while zebrafish is a really nice, well-studied fish, it's not too food relevant when we think about cultivated seafood. There's also a paper from goldfish, and while again, that's not as food relevant, that paper is pretty interesting to the field. It was one of the first cultivated meat and seafood papers published back by NASA in 2002 when they were exploring alternative food production systems. And so before I dive into our research on some of these fish cell culture work, I wanted to briefly touch on some of the reasons why we think cellular aquaculture could have advantages over current fish production. And so when we think about current fish production, there are concerns over food security with just diminishing seafood stocks in our oceans and high waste of product. There's also concerns over food safety with things like antimicrobial resistance through antibiotic use and parasites. And then when we think about the environment, overfishing is obviously deleterious, and aquaculture can sometimes damage land. But one benefit of the current system is that it's able to provide nutritious, relatively low-cost fish source to a lot of people around the world. And when we envision what we think cellular aquaculture will look like, we do think that in terms of food security and food safety, there would be advantages. So we think that in terms of food security, by having a bioprocess that's more controllable, we'd have a more flexible and adaptable system to really vary with supply and demand a bit better. And then in terms of food safety, because we have a bioprocess that can be monitored and we have testing that can be done on batches, we do think that the products will be sterile. I think in terms of environment and cost, that's where a lot of research still needs to happen. So our lab has been working on life cycle assessments to evaluate the environmental cost as well as techno-economic analyses. And while those models are still being built, obviously the better the experiments, the better the data is to build those models, the more promising it will look for that. And so to dive into our research, we in our lab focus on a variety of different cell types. We do mammalian work, I do fish work, Sophie does insect work, and that fish work has led us to some really fun adventures at the lab, including with our PI David. So we've done some fishing trips, as well as collaborations with academic labs, and that's led us to now having 4 different fish cell lines that we use for our work. So the Atlantic mackerel cell lines, we have two muscle Atlantic mackerel cell lines. We term them MAC1 and MAC2. We also have an Atlantic bluefin tuna muscle-derived fibroblast-like cell line, and as well as a channel catfish fin-derived fibroblast-like cell line. And so with these cell lines, we're now looking at how to answer some of those research questions along the cultivated meat and seafood process. And so today I'll just touch briefly on some of the work we've done. When we look at the cell sourcing, all of our cell lines have been spontaneously immortalized, and so we're pretty curious as to what enabled that immortalization. We also are curious about what the best long-term storage method is with cryopreservation, and we've actually had some issues with that recently. And for our proliferation bioprocess, as I mentioned, reducing animal products is going to be very key. So we're developing serum-free media that can allow for the growth of these cells. Differentiation into muscle and fat is going to be important, but that differentiation needs to happen efficiently. So we're looking at different media to induce those phenotype changes. And then finally, when we think about products, I think that most of the first products that you'll see in the market with cultivated meat and seafood are going to be what we call hybrid products. So that's a mixing of plant material with animal cells. And so we're curious about using plants as a scaffold to provide a base for these cells to grow and differentiate. And so the first thing we'll touch on is the cell sourcing and how to study immortalization. And the route that we've taken is by using RNA sequencing to uncover those gene expression changes that can be the basis for this immortalization. And so we did an experiment where we did our RNA-seq of our MAC1 and MAC2 cell lines, both pre- and post-immortalized, as well as we did RNA-seq of different phenotypes of that MAC1 cell line. So proliferating cells, muscle-differentiated cells, and then lipid-loaded cells as a fat analog from these muscle cells. And I think through the PCA you can see that the phenotypes, that proliferation, lipid loading, or muscle, is what accounts for most of the variants seen in our cultures, but we still do see differences between the pre- and post-immortalization, and we even see quite drastic differences between our MAC1 and MAC2 cell lines. And so we're able to use that RNA sequencing data and run analyses like gene set enrichment analysis to look at pathways. And so the figure here shows KEGG pathway analysis. And then on the right of this figure, we see what's increased in immortalization or what's enriched in immortalization with a positive normalized enrichment score, and we can see things that are pretty typical of a more proliferative cell type. DNA replication in cell cycle, for example, are upregulated in our immortalized cells, but then when we look at what's more prevalent in our primary cells, we do see more ECM cytoskeleton focal adhesion processes. And so this is just a snapshot of the RNA-seq data that we're now using in experiments looking at CRISPR knockdowns to induce immortalization. And then the next phase that we're really looking at quite extensively is growth medium and how we can reduce the serum in our growth medium. And so this work comes from a postdoc in our lab Taewon Lim, who had took our MAC1 cells and looked at developing a serum-reduced media for these cells. So essentially what he did is he took our growth media, which was 20% FPS, gradually reduced it down to 2.5%, and started to screen for different components that could help proliferate the cells better. So his first screen was just looking at one factor time screening to identify small molecules, and he identified 7 components: linoleic acid, ascorbic acid, sodium selenate, FGF2, TGF-beta, PDGF, as well as insulin. And he found that those 7 components helped promote cell growth a bit in low serum. But then we started to think about how to replace the albumin that's the most prevalent component in serum. And so we screened different plant and algal extracts, and he found that this rapeseed protein isolate, which is a byproduct of canola oil processing, had significantly improved our MAC1 cells compared to a control with no rapeseed protein isolate. And so he was able to take a lot of his different findings, so those 7 components, and a mixed basal media, that's L15 and F12, as well as with that rapeseed protein isolate. And then you can see in the black bar, that's our control, where we have 7.5% FPS, and then in the triangle tan bar, that's his new low serum media that has 0.1 mg per mil RPI, and he got similar growth. So he was able to significantly improve the growth in low serum, but there still needs to be challenges we still need to overcome challenges to get completely serum-free growth. And then when we look at the differentiation of our cells, one important factor for cultivated meat is that we're really trying to replicate the muscle and fat tissues that we see in our products, and so muscle and fat differentiation are going to be really important. And so if you remember from the RNA sequencing, we did RNA sequencing of our different phenotypes. And so one thing that we're interested in is we could actually look at the differential expression of, say, the muscle and the fat, the muscle versus the proliferation phenotype. We then identified receptors that were upregulated in muscle and added ligands to those receptors to help induce that muscle differentiation. So this is showing one example where we did this. We identified a ryanidine receptor, significantly upregulated in muscle. We identified a couple different ligands, so arginine and S107 ryanidine stabilizer. We found that when we added those ligands to our cell culture, we could get an increase in the percent myosin heavy chain positive nuclei, which was quite significant, especially with arginine. And so you can even see here in our full well images that our control, and this is for myosin heavy chain staining, our control had some staining, but then when we added our 4 millimolar arginine, we got a significant improvement in our differentiation. We've done something similar for our lipid loading and found that LPA, or lysophosphatidic acid, is also really enabling better lipid loading in our cells. And finally, as I mentioned, we think the first products are going to be a hybrid product of plant and animal cells. And one of the main components of meat that isn't muscle and fat is the extracellular matrix. It gives a lot of the structure to our products. And while it would be great to use extracellular matrix proteins in our cultures, most of those proteins are animal-derived, things like gelatin and collagens. And so we have a lab technician and master student, Connor Joyce, who looked at ways to replace the extracellular matrix proteins by actually looking at plant scaffolds instead. So we have a gelatin control, which is fish skin gelatin, but he also explored different plant proteins like zane, which is a corbin protein. He had alginate and chitosan, which are both polysaccharides, and cellulose, which is also polysaccharide, and then soy protein. And he found that the zane, the soy, and the chitosan did really well in promoting the MAC2, the second macro cell line growth, as you can see with the DAPI, the phalloidin, and the myosin heavy chain staining. And so these were all done in film, so a 2D culture, but now we're scaling these up to scaffolds to see how we can make 3D cultures with our fish cells. And so I've just given you like a really high overview of what we've done in the lab. We definitely have a lot more experiments that I don't have the time to share, but I'm happy to answer any questions or talk after this about these. And what I've shown you today is cellular agriculture for a really well-studied characteristic species that we're used to eating. Now Sophie will tell you about some more non-characteristic species.
Okay, is this working? Okay, great. So yeah, as you can guess, our unconventional cell source is insect cell meat. So I'm very excited to talk to you about some recent research in our lab with these cells. And so bringing back this graphic from Michael, as he said, most of the research in our field focuses on these traditional cell sources, so cows and pigs and chickens. But when we're growing meat from cells instead of animals, we're no longer constrained by species that we can easily domesticate. And that's why we're eating cows and pigs and chickens, because they're easy to domesticate. And instead, if we're thinking about growing meat from cells, we could look for cells that more easily meet the criteria of cell-cultivated meat, because it's a really challenging thing to grow. And by these constraints, I mean cells that are low cost, scalable, and of course, animal-free. And it turns out that insect cells, as a lot of you probably know, easily meet these criteria, especially when compared to these mammalian and avian cell sources. And so that idea is how the tobacco hornworm made its way onto this graph, and what's got our lab excited about studying these cells for a food product in the past 6 to 7 years. And these are some of our earlier research on the topic. And I'm sure, I guess, as this is a lot of insect cell professionals in this audience probably know what I mean when I say insect cells are low cost, adaptable, and robust. So bear with me if this is kind of very basic knowledge. But to give you an idea of what the first experiment that got our lab excited about growing these cells for food was before my time with a former graduate student named Dr. Natalie Rubio. She started her PhD working with fruit fly muscle cells. This was just what we had at the time. She was culturing some of these cells. And in something that I'm sure is probably familiar to a lot of you that work with cells, she forgot about them. And she went on vacation. She came back a couple of weeks later, realized her mistake, and thought, well, my cells are probably dead, but I'll just check them. And many of you might not be surprised to know that the cells, instead of being dead, were actually thriving. So she thought this was really interesting. Maybe I'll repeat that as a more controlled experiment. And so she did that. She saw what happens when you intentionally starve fruit fly muscle cells over 25 days, so just don't give them any media, and compared that with the mammalian cell source. And what we had at the time was mouse muscle cells. And you can see she saw the same thing. After about 15 days, so the green there are live cells and the pink are dead. After about 15 days, the mouse muscle cells are starting to struggle. By 25 days, they're all dead. The fruit fly muscle cells, on the other hand, are doing totally fine. So when we're trying to create cells for food systems that are simple and scalable and low cost, starting with cells that require fewer inputs is a great start. And besides just allowing us to continue research when we're on vacation or at a conference in St. Louis, insect cells have a lot of other really important traits when we think about growing them for food. To try and demonstrate that, I've put together a kind of cultivated meat wish list, which is what a lot of companies and researchers in the field look for when they're establishing cell lines for meat. And we'll go through and see if insect cells can meet these criteria. And before I get into that, most of the cell lines that I'll talk about come from Manduca sexta embryos. This is just cells that our lab has worked on establishing. So this is just our basic protocol for getting cells. Manduca sexta is a caterpillar, turns into a moth and lays eggs. We collect those eggs, sterilize them, release the cells from the eggs mechanically, get rid of the debris, and then different plating paradigms can leave us with different cell types. So our first wish list item is immortalized cells. So very similar to Michael's cells and similar to fish cells. If you work with them, insect cells are just known to be easy to spontaneously immortalize just by continuous passaging. And this is still a challenge for many mammalian cell lines, especially in those livestock species. And so you can see this is just one example of a cell line established in our lab from those Manduca sexta cells. Unsurprisingly, after we kept passaging them, we got a homogeneous cell population that reached over 100 doublings, and we've been able to culture this as well as other cell lines for multiple years in continuous culture. So number one, check. Next is growth in animal-free culture media. Michael showed some data with fish cells that this is still an active challenge to remove animal products from the process. course, this is, again, really important to us because we want to take animals out of the meat production process. And if you work with insect cells, you probably already use serum-free and animal-free media. And that's because the industry has solved this challenge decades ago. And insect cells are known to be very adaptable to different media formulations. And so, again, just an example of our cells being adapted to commercial serum-free media. We don't have to do any optimization in lab because it already exists. And this is just over 4 passages. We decrease the serum. We usually use serum when we isolate cells because that's still a challenge to remove it at that point. But we've been able to grow continuous cell lines in just serum-free media, as I'm sure a lot of you do as well. So number 2 is check. Next is robust growth in single-cell suspension. This is favorable for cultivated meat because we're looking to create kind of unprecedented amounts of cell culture. And suspension culture is favorable to adherent because in adherent culture, you're constrained by surface area. And when we're growing cells in suspension, we're constrained by volume and how many cells we can pack into that flask. So it's a lot more scalable. And single-cell suspension is favorable to using microcarriers because we don't have to worry about microcarriers that are edible or environmentally sustainable and things like that. And so insect cells, again, are often known to grow really well in single-cell suspension. This is an adaptation procedure that our lab used to generate a non-adherent cell line. We started with a mixed population where we had some cells stuck to the bottom and some cells floating. Over 6 months, we just passaged the floating cells until we got a really robust non-adherent cell line. In many of the traditional livestock species, cells are still adherent, and when they're grown in suspension, often have to use microcarriers. So this is very favorable. Once we got this really stable cell line, we wanted to see how many cells can we pack into a unit of media, because again, we don't only need to worry about the components that go into our media in terms of cost and environmental sustainability. We also need to think about the volume of media that we use. So how often do we have to feed our cells, and how many cells can we pack into a unit of media? And we saw for this cell line that I showed the establishment procedure before this, without any optimization, they reached a relatively high density, especially again compared to the mammalian cells, where they reached over 25 million cells per ml without any further optimization, and without media exchange, they could reach about 4 million per ml. We also wanted to see how easily they could be transferred to different culture vessels, and they were easily grown in both shake flasks and 2.4 liter airlift bioreactors. And if you don't work with suspension cell culture, just to demonstrate what it means by really high density culture, this is just showing our shake flasks without cells, and then when we add cells and grow them to that high density, they get so dense that it becomes a completely turbid, almost opaque cell suspension, to the point where often people will tell you that your cultures are contaminated, but I promise you that one is not. It's just really dense insect cells that we can then, when we spin down, we get these really big cell pellets on the order of grams, which may not sound like a lot in terms of meat production, but at the lab scale, it's really exciting because we can then take this material and study it in more of a food-relevant context instead of just under the microscope. So check on that. And then finally, it's great if we can culture cells in low-cost, animal-free media, high-density suspension, but none of this matters if we don't want to eat them. So we next need to study, are these cells viable food ingredients? And currently, the only FDA and USDA approved cell lines for consumption are from 2 companies, Upside and Good Meat, who use embryonic chicken cells. And so we use those as a comparison to put ourselves in context. DF1s are embryonic chicken cells similar to what these companies use that we grow in our lab. We compared these to our Manduca sexta non-adherent cell line, or MSNACs, which we call them. And you can see morphologically, the cells look very similar. We then wanted to compare the nutrition of our cells. So there's published data in the safety documentation of these companies with their embryonic chicken cells. We compared this to our MSNACs. Again, an advantage of being able to create such a big cell mass is we can send our cells off to kind of traditional food characterization companies. We saw that the dry weight proximate analysis between our MSNACs and these company cells are very similar. We went a little bit further and looked at amino acid and mineral breakdown of our cells compared to chicken cells. We saw that for the amino acids, we're also comparing to both to the cultivated chicken, which are the blue lines, and then these companies also published conventional chicken data. And we could see that the amino acid breakdown was kind of surprisingly very similar between our insect cells and these chicken cells. And then on the mineral side, the insect cells had slightly more iron, calcium, and zinc. So this is still very early. We're not ready to just feed you all insect cell embryonic biomass yet, but we thought this was a very promising start for the nutritional data. We're also interested in muscle, just like Michael was showing. So we work with some Manduca sexta muscle, as well as we're exploring Echidna domesticus or cricket muscle. People are a lot more familiar with eating crickets. You can see, this is probably familiar if you've ever done embryonic insect isolations. You get these really nice contractile muscle bundles. When we isolate the muscle and separate it, we can get these really beautiful myosin heavy chain positive striated muscle fibers. However, we're still struggling a bit with the proliferation and control of these cells. So if anyone here works with insect muscle, please come give me advice. But we're still working on those. And then finally, fat is really important. Michael mentioned fat's important for flavor, texture, nutrition of meat. So we did a study where we wanted to turn our Manduca sexta embryonic cells into a cultured fat source. We borrowed a method that's been shown to work in other cell types where you just feed the cells intralipid, which is a soybean oil emulsion. You can see here, the more intralipid we add, the more fatty the cells get. The red are lipid droplets. And so we were able to get these really fatty, cultured insect fat sources. And then in this paper, we did some further characterization of fatty acid profile and other meat-relevant properties that I don't have time to show. But this is what our insect fat looks like. Very, very appetizing, I'm sure, to everyone. So that's just, again, a snapshot of some of our lab's work in insect cell culture. Is very early stage. I think what gets us really excited, beyond just these basic properties of insect cells that make them favorable for cultivated meat, is the potential to expand. As we know, insects are the most evolutionarily successful group of organisms with an incredible range of species diversity. It's possible we could get cells from new species that are tastier or more nutritious or have interesting growth properties that we have yet to explore. So if you work with insect cells for a different application, you've always looked at your cells and thought they look really tasty, feel free to come talk to me later. But yeah, that's our presentation. I don't know if we have time for questions, but happy to answer some.
All right, we do have 5 minutes for questions, so I will.
Yeah, thank you for sharing. By the way, I have been eating insects from locusts, locusts. Have you eaten cockroaches? Yes. Yeah. Then the abdomen, and then crickets, beetles, and of course silk moth pupa. But apart from that, in one of your last slides, you say something about accumulation of fatty acids. Is it only accumulation, or is your cell culture system also providing to do, let's say, synthesis or modification of fatty acids in a specific way? But I have no idea about your system of these fatty acid biosynthesis. But for instance, I know that insects have the potential of doing modification of fatty acids because they have the genes. So is this a property that could be explored as an interesting asset?
Yeah.
I have no idea, I mean, because you talk about accumulation.
Yeah.
Accumulation is in fact not interesting because you have to put your fatty acids into culture medium and then your cells absorb. But if you will have modification biosynthesis, that would be quite interesting.
Yeah, exactly. Thank you for your question. So in that paper, we did lipidomics, where we could study the fatty acid breakdown, and then we can compare that to just plain intralipid. And we saw that a lot of the lipids match up, but we do see increased lipid or fatty acids that are not present in intralipid. So I think it's likely that they're making some of their own, but we need to study that a bit further.
I think up here, we had our next question hand up go up quickly. So
Thank you. This is very interesting. We're from like plant science background, so this is pretty new. So I had a question about what serum-free meant.
Yeah, so most of animal cell research uses fetal bovine serum or other animal serum. And so serum-free just means culturing without that serum.
So I have two questions actually. So the first one, I was like wondering, so like when choosing the insect that you decided, how was that like process? Because there's like hundreds and hundreds of millions of insects. So how did you manage to like narrow it down essentially to that one?
Yeah, good question. So we used the tobacco hornworm just because we had access to it. There's a lab that's next to us that has a colony of Manduca sexta. So that was just out of convenience. When we're going into new species, a lot of the times we're looking at species that people are more familiar with consuming. So crickets, we're also interested in mealworms. So yeah, mostly convenience, and then ones that people are interested in eating in the future.
Okay, that makes sense. Thank you. And then I guess the second question I had, so I know you talked about commercializing fish in your presentation, and I was kind of wondering, like, do you have any specific, I guess, like, plans on how you were able to, like, potentially commercialize it? Because I know, like, right now, especially Beyond Meat and those products, they're very expensive, and so I was, like, wondering how potentially some of the methods you were potentially thinking about to, like, reduce the cost so that way it's more affordable, especially those that are on a limited income, for example, because I know the goal is to make it more sustainable so that we don't have to rely on fish in the future.
Yeah, thank you for that question. I think that's a big question for the field in general, and while we don't focus on commercialization, we do have perspectives from the field. I think one thing that people will do early on, especially as these bioprocesses are pretty expensive, is to lean towards hybrid products. So while plant-based meat is good but doesn't taste great to most people, we do think that adding an animal component can really help improve the flavor of plant-based meat. And then we just need to rely on innovations like alternative sources that scale better, like novel bioreactors. And so I am giving a talk on Monday on a bit more of a larger perspective of the field, where I'll touch on some of the innovations that are being made there.
I think we have time for one more question. I thought there was one, maybe.
Okay, it was very interesting talk. I'm just curious about, and I need your thought, because this process itself, when you talk about scaffold to make entire meat from a cell, I'm wondering, because these cells could be bioreactors to make proteins itself. So why you have to go through that long process to make a meat instead of just making a protein of nutritional value to provide people? What is your thought for that?
Yeah, and so I think, again, I'll talk about this more on Monday morning, but when we think about cellular agriculture, we think about two types of products, acellular versus cellular. And so there are efforts being made to produce proteins, like milk proteins, egg proteins, even muscle proteins, in species like fungus, E. coli, or fungus, bacteria, yeast. And I know that insect cells as well are used extensively in protein production.
Great. Thanks. Well, I think we are now at break time. And so if you can join me in thanking both of our presenters today.